Hello, we are Yebin Kwon and Sangmin Oh. Today we're going to present about our final assignment in molecular biology called Transcriptional Regulation in the Lac operon, especially in lambda phage and Drosophila. Please enjoy our video and let's move on to directly on theme 1, which is transcriptional regulation of lac operon.
The second topic is transcriptional regulation of lambda phage, and I'm Yebin Kwon, explain on this topic. Lambda phage was discovered in 1950s by Lederberg. It is a bacteriophage that infects E. coli and has played a key role in molecular biology research. Lambda phage contains approximately 48.5 kilobase pair of linear double-stranded DNA, which circulates inside the host cell after infection. Its genome contains critical DNA sequences that determine the phage life cycle. Lambda phage has two life cycles, the lytic cycle and rhizogenic cycle. Which cycle is chosen depends on the environmental condition of the host cell. In the lytic cycle, the phage replicates inside the host, lyses the cell, and releases new viral particles. In the lysogenic cycle, the phage DNA integrates into the host chromosome, and during cell division, the prophage DNA is passed on the daughter cells. There are 4 key genes regulate these life cycles. The first one is C1, also called the lambda repressor, which acts as a rhizogenic activator. The second one is C2, also a rhizogenic activator. When both C1 and C2 are strongly expressed, the rhizogenic cycle is promoted. The third one is C3, which protects C2 from protease-mediated degradation. Finally, CRO, the master regulator of the lytic cycle, acts as a repressor of C1 production. In summary, strong expression of C1, C2, and C3 leads the rhizogenic cycle, while strong expression of CRO promotes the lytic cycle. The level of C1, C2, C3, and CRO are determined by the promoters near their genes, which include PL, PR, PRM, and PRE. In the rhizogenic state, PRM is on, while PL and PR are off. In the lytic state, PL and PR are on, while PRM is off. In more detail, in the rhizogenic state, C1 binds to operator 1 and operator 2, repressing PR and PL, while RNA polymerase binds to PRM to transcribe C1. In the lytic state, CRO binds to operator 3 and represses PR, allowing RNA polymerase and transcription machinery to bind PR and PL. This difference arises from the opposite operator binding affinities of C1 and CRO. Based on the E. coli growth curve, let us consider phage infection at point 1 and 3. Point 1 represents a nutrient-rich environment with abundant glucose, favorable for rapid E. coli growth. Point 3 represents a nutrient-depleted environment with both glucose and lactose exhausted, where cell growth is minimal and cell numbers remain stable. At point 1, lambda phage favors rapid replication and infection of neighboring cells, leading to the lytic cycle. CRO binds operator 3, represses PRM, and RNA polymerase with sigma factor binds to PR and PL, initiating transcription. The N gene is expressed, allowing RNA polymerase to read through CRO, Q, and other genes to ensure their expression. At point 3, with poor growth conditions, the phage prioritizes survival and integrates its DNA into the host genome, leading to rhizogenic cycle. C1 represses PR and PL while activating PRM, establishing lysogeny. Here, C1 binds to operator 1 and operator 2 represses lytic promoters and allows RNA polymerase to transcribe from PRM. Through this process, lambda phage infection and host cell life cycle gene regulation are coordinated, enabling the phage to implement an optimal survival strategy. Now, let's move on to the last topic, combinatorial transcriptional control of eukaryotes. Lastly, the question 3.4 is to design a new enhancer that drives the highest gene expression at the position between the 2 points, box 2 and box 3. In this case, the concentrations of the transcription factors cannot change, but the transcription factor binding sites and their binding affinities can be reorganized. The position of the stripe peak is determined by the relative balance between activation and repression. If activation occurs more anteriorly than the current state, the stripe peak appears further to the left. If activation occurs more posteriorly, the stripe peak appears further to the right. Therefore, we propose two enhancer design strategies to drive expression more posteriorly. The first is to increase the bicoid activation threshold. This can be achieved by lowering the affinity of binding sites and reducing the number of bicoid binding sites. The most frequently occurring bicoid consensus motif is TAA-TCC. And the more the if gene sequence matches this motif, the higher the binding affinity. Therefore, the sequence in the if gene that matches this motif can be completely altered to reduce the number of binding sites. Or, the core motif can be mutated, for example, from TAA-TCC to TAA-CG, to lower the affinity. The second strategy is to strengthen giant repression while simultaneously weakening crupo repression. Currently, at the beginning of box 2, giant acts weakly, and at the end, crupo repression is strong, producing a stripe within the box. To shift the stripe posteriorly, giant repression should act more strongly than in the current state, and the crupo repression should act more weakly. In practice, this can be achieved by increasing the number of giant binding sites or modifying its consensus motif to raise affinity. While for crupo, the number of binding sites can be reduced or core nucleotide can be changed to lower affinity. So far, we have explored the mechanism of transcriptional regulation in log operon and lambda phage, as well as combinational transcriptional control in Drosophila. Before we conclude, I would like to express my sincere thanks to my teammate Sungmin for his contribution and support throughout this project. We would also like to extend our thanks to Professor Kim for providing us with such insightful lectures. This concludes our presentation. Thank you.